Okay, this is video number 6, and we'll talk about post-hoc test and reporting results of two-way repeated measure ANOVA. We found there is a significant two-way interaction between time and treatment in self-esteem scores. In this case, post-hoc test can be done by run repeated measure one-way ANOVA of the first factor, treatment, at each level of the second factor, time, for this case. We ran ANOVA underlying test function grouped by the time variable. Considering the Bonferroni adjusted p-value, p dot adj at the last column, the main treatment effect was not significant, meaning the mean self-esteem scores are not significantly different from the control between control and diet groups at the time 1 with adjusted p-value of 1. It becomes significant at time 2 with the adjusted p-value of 0.036 and time 3 with adjusted p-value of 0.00051. In other words, pairwise comparisons show that the mean self-esteem scores were significantly different between control and diet groups at time 2 and time 3. We can also swap the first factor and the second factor and run repeated measure one-way ANOVA. At this time, we grouped by treatment and ran one-way repeated measure ANOVA for time. The result shows that the effect of the time is significant only for the control trial, with the adjusted p-value of 0.0000001. Pairwise comparisons show that all comparisons between time points were statistically significant for control trial, but not for diet group. If the interaction is not significant, we can run main effect tests using pairwise underlying test function. In the self-esteem example, the interaction is significant, thus the following steps are not needed. Here, we are just showing an example of running post-hoc tests for the main effect for the future reference. If there is significant treatment main effect, the first code chunk will be appropriate. If the time effect is significant, the second code chunks can be used. Once more, we want to present the meaningful results on one figure. Using the similar code, we will generate box plots with pairwise comparison results written on the top of the box plots. The first 3 codes, line of codes, is exactly same as the pairwise comparison codes at the previous slide. Now we just call it PWC, so we can use the pairwise comparison results on the plot. To generate box plot, we are using ggboxplot function in ggpubr package this time. Again, as similar as before, we set expedition at time to add the post-hoc results PWC to the box plot by adding add underline xy underline position function. Then we add stat p-value manual parenthesis PWC function to add pairwise significance on the box plot. We also add subtitle and caption with lab function. labs function. This code generates the following figure with 6 box plots for time and treatment combination. We'll also add the pairwise comparison results on the top of the box plot. For time 1, beginning of the study, diet and control group self-esteem scores are not significantly different. For time 2, the mid of the study, diet and control group self-esteem scores are significantly different, with 1 asterisk mark there. For time 3, the end of the study, the treatment and control, the diet and control group self-esteem scores are even more significantly different, with the 3 asterisk mark here. Again, the repeated measure ANOVA results are on the subtitle, and the pairwise comparison labels are on the caption. This is the end of Module 4 lecture.